Today we will be talking about the ECG or the EKG, the Dutch or German version. Basically, it is an electrocardiogram. Now, before going into detailed lecture about ECG regarding the nomenclature or the terminology in ECG, it's better if we know some basics of ECG. Now, by definition, what is an ECG? We know every living tissue or every living being, a particular characteristic is not absolute, but a major characteristic, it's being mobile or showing some motion. Now, zooming into this tissue of a living being, particularly at subatomic level, we also expect movement of its subatomic particles like electrons. And in physics, when we see movement of such uh, electrons, we coined it the term electricity. Now, of course, the same electricity can be present in the heart, and an ECG is nothing but reading of this electrical activity of the heart. So now if we simply define what is an ECG is nothing but a galvanometer reading of this electrical activity of the heart. Now, what makes the heart tissue special? Of course, heart is a muscular tissue, but what makes this cardiac tissue different or unique from the other muscular tissue, particularly your skeletal tissue or your smooth muscle tissue? It's the presence of the intercalated discs. The intercalated discs are one of the main reasons why your heart has to beat non-stop from the time you're born till you face death. They are one of the reason why your heart muscles, without relaxing even for a bit, or better to say without stopping for a bit. Now, zooming into this intercalated discs, we will find at histological level the organelles which are desmosomes or gap junctions. By nature, desmosomes, they have more adhesive characteristic which joins or holds the cardiomyocytes strongly together, and the gap junction is they are nothing but channels for calcium. Now, calcium, they are used by your myofibrils for contraction, and eventually this calcium has to go back into the sarcoplasmic reticulum. But some of this calcium moves into its neighboring cell, moves into the neighboring cell via this gap junctions, and this is how a wave-like excitation moves from one cardiomyocyte to the next cardiomyocyte. And this is the reason when the heart contracts, all muscles contract at the same time, and they also tend to relax at the same time. Now, keeping this special criteria of the heart muscle, there are also some other heart cells, particularly in your endocardium, the endothelial cells, the in your basement membrane, the fibroblast, then your smooth muscle cells in various vessels. But what is important for ECG is the presence of ventricular and atrial cardiomyocytes and your conductive cells, which also include a specific type of cells called your pacemaker cells. Usually, the activity in the ECG, the activity of cells that are responsible for the different patterns in an ECG is mainly due to these types of cells. So any ECG changes which we see during pathological condition usually results from such cellular changes in the mentioned cells, that is pacemaker cells or your electrical conductive cells or your myocardial cells. So at the end of this session, I would conclude by talking on the last topic of physiological action potential. Now, before going specifically for the pacemaker and cardiomyocytes action potential, let's revise a bit of cellular action potential. What does this topic cover? Now, usually when we see the electrical activity in a cell, or when we refer to the electrical activity which occurs in a cell in terms of excitation, we usually see the activity near the cellular membrane. That's why the baseline that we see over here, it's also called the resting membrane potential. Now, usually this in physiology we determine or we denote in the form of a graph in which the y-axis is a voltage and the x-axis is time. Now, when we plot a graph, we see, don't freak out if you see such a pattern. It's quite easy when you try concentrating at it. Now, the resting membrane potential you can see over here is minus 70 millivolt. Of course, there are stimuli which tends to increase the charge or which tends to make your cell more positive, and this you can see in such fluctuation. This as we move up towards the graph, it becomes positive, and such stimuli tends to raise the voltage. And at certain moment you will see that the stimuli has been successful to meet the threshold potential, and it is at this potential that there are voltage sensitive channels, particularly voltage sensitive sodium channels, which open. And we know the sodium concentration outside the cell is relatively higher compared to the inside. This is, you can consider this is one of the reason which corrects the hypothesis that we all, our living form came from the ocean. Anyhow, keeping this philosophy aside, so naturally in the human body the sodium concentration outside the cell is usually higher, and with the opening of the voltage gated sodium channel, sodium tends to move following its concentration gradient into the cell. And as sodium rushes into the cell, and you can see with respect to time, with a very less amount of time, the cell's voltage difference, it shoots to plus 40 volt. And this point, or this potential of action of the cell where it is limited, this is also called the action potential of the cell. It is at this point, if we look at the cellular membrane activity, where the sodium channel or voltage gated sodium channel closes. And after this point where they are closed, voltage gated potassium channels are open. Voltage gated potassium channels, when they are open, when the voltage gated potassium channels are open, again based on the concentration gradient, potassium concentration tends to be higher inside the cell relative to outside. That's why potassium tend to move out of the cell. And since the potassium moves out of the cell, positive charge of voltage is now being lost, and slowly the cell starts becoming less positive and moves towards the negative. Usually by nature, compared to the sodium channel, potassium channels are slower, and that's why you see the graph moving more and down. And this phenomena is called hyperpolarization. And this hyperpolarization, it's compensated by the activity of pumps. The pumps use ATP as a source of energy, and this brings back the membrane potential to its resting state, where the pump uses ATP to pump 3 sodium out and 2 potassium into the cell, and this restores back the resting state. Now, for ECG, it's not necessary to memorize all the story that I have just said you, but just know what happens during depolarization, what happens during depolarization, or what is depolarization, what is repolarization. Mainly these are the main two terms you have to know. And if I say you an action potential, it's not necessary for an absolute definition. Now, if you want a definition for depolarization or repolarization, certain textbook mention them, and I can read it out to you. Uh, Cardiac action potential is a result of a sequential flow of electron across ion channels in a cardiac cell membrane. This results in electrical activation of myocardial cells. Now, what depolarization is defined as the movement of cation into the cell, producing positive cell charge relative to the outside. Repolarization is quite a vice versa. Cation movement, particularly potassium, out of the cell, reducing the positive cell charge. Now, out of this all definition, what you need to understand is what does depolarization mean, or what does repolarization mean? Because out of everything said, these are the main to take away from this lecture. Going further into cardiac action potential. Cardiac action potential at cellular level, we can see mainly two types of cell. One is in your pacemaker cell and the other in your cardiomyocytes. Now, both in pacemaker cells and cardiomyocytes, we can see particular characteristic common in which the electrical event that takes place is divided into certain phases from 0 to 4. You can see in pacemaker cells they lack phase 1 and phase 2, whereas cardiomyocyte has all four phases. Now, the voltage changes, or the dynamic in the voltages with respect to time, is a function of the working of the channels of the membrane. Now discussing each phases, we can see what happens. Now, during the phase 4, during the phase 4, the sodium channels open, the sodium channel open, and these channels are sensitive to hyperpolarization. When the cell is hyperpolarized, the pacemaker cells gets hyperpolarized, the sodium move into the cell, and the channels are so called funny channels. I have no idea why the scientists called it funny channels, because it's not so humorous among many students. Anyhow, going further, and this movement of sodium through the funny channels slowly depolarizes, brings the membrane potential to its threshold potential. Again, as discussed, if the threshold potential is reached, there are voltage gated channels. Now, voltage gated channels over here are your T type and your L type calcium channels, which open, and there is a strong, very strong inward influx of calcium with its concentration gradient, and this leads to a rapid depolarization, which is also marked as phase 0. And at the end of this activity, or when the action potential is reached, the phase 3 starts. Now, whereas phase 1 and phase 2, it's usually absent in pacemaker cells, and what comes next is phase 3. Now, during phase 3, as we have seen already, in a normal cellular membrane, there is opening of potassium channels. As potassium moves out of the cell loses its high potential difference or membrane voltage, and slowly it decreases until it's hyperpolarized, and again phase 4 is continued. So this is something that occurs in a pacemaker cell membrane.
Dr. Andrews said smiles are contagious, and I'm testing that hypothesis.
Well, I'm not sure your study will prove anything. Might have something to do with the lack of the Duchenne marker.
Oh, yes.
But here you go.
That wasn't a smile.
Sure it was.
I want to show you something. You need to follow me.
Okay.
The imposters. That's a little bit unsettling, Murphy.
Why are you studying the current of ions during electrolysis?
It's not the chemical kind of transference. It's the doctor-patient kind. I'm dealing with a personal situation.
I don't think the internet is very helpful for solving personal situations.
True.
Captain Murphy? It's your mother's smile.
She seemed like a good template.
There is no Duchenne marker. Her smile is not real. All the other ones are. All the other ones are.
A cardiomyocyte is somewhat different in terms, as in phase 0 there is the rapid influx of sodium into the cell. This inward current is responsible for rapid depolarization. Then there is a particular characteristic also called as the plateau phase. During this phase there are two things that happens. One is which makes it different from pacemaker cells. First, the potassium channel open, and that's why you can see a drop in the voltage, and also over here the L-type calcium channel open. The L-type calcium channels was also present in the pacemaker cells, and these are the one that are responsible for phase 0. And the calcium channels which are present over here, again they tend to stabilize, or they maintain a certain potential as when the potassium moves out. But after a certain period, these calcium channels or L-type calcium channels are inactivated, but the potassium still tend to move out. And this phase 3, like the pacemaker cell, is the repolarization phase, and this continues till the potassium channel reaches an equilibrium between the movement of sodium and calcium in and out of the cell. And after this, the cycle is followed up. Now when we compare both of them, you can easily see the morphologic differences between the graph of a pacemaker cell and a cardiomyocyte based on the working of different types of channel at different intervals of time. Now, before finishing off this topic, it's better to discuss one last topic of refractory period. Now, what is refractory period? Now, by definition, refractory period is a time during which a cell performing a function, is relieved from its duty. Now, just to make it more simple, I can give you a basic example of refractory period is a post-coitus event, or post-ejaculatory event. Now, during this event, what happens is after the male ejaculates, he needs a certain period of recovery to, how do you say, get into mood. Now, this period can be somewhat analogous to the heart refractory period, where the heart is a bit stubborn regarding to responding. Now, again, refractory period can be further classified into absolute and relative. Now, absolute refractory period is the period when even after giving a stimuli, a very strong stimuli, the cardiac muscles the cardiomyocytes do not tend to respond, whereas relative refractory period, it's a period that applying a very strong stimuli, the cardiac cell can be excited once again. Now, absolute refractory period in a graph, it usually lies at initial 1/3 of the cardiomyocytes action potential graph. It's usually from here to here. And if you look at the membrane, this is due to most of the channels, they are gated channels. That means they have some gates here and here. And when I say they do not respond, that means the sodium or the cation cannot move in because they are gated channels, and these gate, usually when I say they are inactivated, they tend to remain closed, and they need a recovery time, or they need a period to come back, or where this gated are opened up back. During an absolute refractory period, these gates are completely closed, and that's why the sodium does not flow in and they remain refractory. But after a certain period of time, slowly the number of these inactivated gates reduce and they open up, and this period is marked by the start of the relative refractory period, where the number of inactivated channels are slowly activated. And over here, it's that a very strong stimuli can move the heart out of its refractory and again start an action potential. Why am I mentioning all this? Because in certain pathological condition, particularly in arrhythmias and all, such phenomena can occur when stronger or an ectopic stimuli tends to increase the excitation of cardiomyocytes, and that's why you will see in arrhythmias sudden increase in heart rate, paroxysmal activities and all. So this was some word regarding refractory period.